I will take a few minutes to introduce our next speaker. So our next speaker is Dr. Avi Maayan. Dr. Maayan is the director at Mount Sinai Center for Bioinformatics and a faculty member at the Icon Genomics Institute. The Maayan Laboratory applies ML to study how intercellular regulatory systems function as networks to control cellular processes such as differentiation, apoptosis, and proliferation. In this keynote session, Dr. Maayan will delve into the transformative impacts of generative AI and LLMs in the analysis of gene sets. He'll discuss how these innovations are enhancing gene annotation prediction, ultimately leading to the discovery of novel therapeutic targets across a broad spectrum of biomedical research applications. The floor is yours.
Thank you, Maya, for the introduction. Let me share my screen. Okay, I hope you all see it. So first, I would like to thank the organizer for the invitation. It's really nice to see how Lucidata has grown and is doing really well. So my name is Avi Maayan, and I'm a professor at Mount Sinai, and my lab is developing software systems and methods for computational biology. And today I'm going to talk to you about 3 projects that are ongoing projects in the lab that could be interesting to inspire you about how to use generative AI in your projects. So the first the name of the first project is called RamaGene, and RamaGene is a project where we mined thousands of gene sets from PubMed Central, supporting materials. The second project that I'm going to mention is called the D2H2, which is a data hub for generating hypotheses for diabetes. And the third one is an older project where we converted the L1000 links data into RNA-seq-like using deep learning and a generative AI. So those names sounds a little cryptic right now, but hopefully in the next 15 minutes, it will become a little bit more clear. So the first project, RamaGene, it's a very recent effort. And for this, we created this crawler that goes through the 5.5 million PubMed Central articles that are publicly available. And it goes through the supplementary materials of those articles. And it's scanning to see if those articles contain human gene sets. And we found about 122,000 articles that have at least one gene set in their supporting materials. And in total, we have almost 650,000 gene sets that we are able to identify in those articles. So some articles have multiple gene sets in their supplementary materials. And then we brought in all of those gene sets into a database that is now searchable. So you can put in your gene set that you found in your study, and then you can get matching gene sets that are in the database. So why do we need RamaGene in the first place? So first, some of the most important findings are actually in those supplementary tables. And all of this data is not searchable before RamaGene existed. So, the idea is that we brought the gene sets that are in those tables to surface for the communities to be able to search through them. We also published a paper in bioRxiv a couple of weeks ago, described the database and the software, but also does some analysis that I'm going to discuss about all of those thousands of gene sets that we were able to extract. So these are some figures from the paper. So on the left, you can see the average size of gene sets that are contributed to the database every year. And you can see that somewhere in the 2000s, there was a jump. And this is probably due to the introduction of proteomics and transcriptomics and other omics assays that really spurred from the sequencing of the human genome. And then it's sort of like stabilized at that level of average 150 gene sets. And this is a combination of short gene sets and long gene sets. We also see that in the last 5 years, there's a huge jump in the number of gene sets that you find in papers. This is also due to the amount of papers available on PMC, PubMed Central, but also there is sort of like a shift in the field of studying gene sets instead of studying single genes. This is a UMAP visualization of the entire RamaGene database, and you can see that there are a lot of small clusters that are probably reflecting a different biology or different pathways and cell types, and also possibly assays. And here we colored each point. So this is a zoom in for most of the points. I think it's 90% of the points in the database. So it's about 500,000 points. And on the left, we colored it by citations, the average number of citations per gene set. So the darker colors have less citations. And on the right is the length of the sets. So you can see that the long lists have less citations. That's sort of like expected, because longer lists come probably from omics experiments, and those have genes that are less studied. So there's a lot of short lists that have highly studied genes and proteins, and long lists that have more understudied genes. You can also do this really useful exercise where we search the table titles for named entities. For example, we can search for kinases, transcription factors, cell types and tissues, or the word up or down to find signatures. And then we can create those subsets of gene set or gene set libraries from the collection of RamaGene. And these are really useful, for example, for doing a transcription factor enrichment analysis, or for identifying the cell type from single cell RNA-seq, or for looking for signatures or perturbations for comparing like RNA-seq study to what is available in the literature. So this is an example of how you can put in the table title search a name of a transcription factor. So here I put in STAT3, and I got 104 gene sets that have STAT3 in the table title. And then you can do that for all of the transcription factors and create a gene set library of transcription factors. And this is what we did recently. We added a transcription factor RamaGene gene set library to Enricher. And you can come here and find the transcription factors that are most likely regulating differentially expressed genes. So here we just demonstrate how, for example, if you do the term search or the metadata search in Enricher for the word dexamethasone, which is a drug that is known to bind to the glucocorticoid receptor. And if you put in the down genes for that drug, you get the second entry in the table of RamaGene, you get the glucocorticoid receptor, which is the known target for the drug. So that shows you that, at least in this example, the RamaGene database that was automatically generated can identify the transcription factor that is most likely the target of this particular known drug and having its known target. So we did that systematically. Basically, we took the RamaGene transcription factor gene set library and we compared it to experimentally determined transcription factor targets from various other sources. And we showed that indeed you get the right transcription factors using the RamaGene gene set library. And this is another chapter in that paper that we just published. So now it's starting to get interesting. And here's where the generative AI comes in. So the next question that we asked was that, can we find gene sets in the database that are really similar? So they have a lot of genes in common, but completely dissimilar abstracts or complete papers. And what we found is that a lot of those gene sets that have a very high similarity and zero similarity at the abstract level are all coming from proteomics studies. So we are able to quantify this by having a list of sticky proteins or proteins that come a lot in the proteomics profiling of cells. And those are typically large proteins that are detected very easily in proteomics and are also highly expressed genes. So this is a little bit of an artifact and also something to say to the field that proteomic studies may be publishing less discriminative types of gene sets. Nevertheless, we were able to create this pipeline that use GPT-4 to try to build hypotheses that once you have pairs of gene sets that have very high significant overlap and also very low abstract similarity, we can give those two abstracts to GPT-4 for hypothesis generation. We also want to check that the two terms, for example, if it is mentioning a gene or a disease or an association between a gene and disease, those terms are not returning any literature. So those are novel hypotheses. So GPT-4 does a pretty decent job trying to make a connection between papers that are dissimilar but have high similarity at the gene level. So this is the results. This is the first entry in the results. And here it found a paper that did the mass spectrometry analysis from the CPTAC profiling of human cancers. And the paper basically identified genes that are correlated with aggressive tumors. And specifically, the gene set that we found to be overlapping with this other paper came from glioma. The second paper is coming from a pull down of this protein called the CLUH or CLU. And that protein is pulling down with a lot of mitochondrial proteins. So what we find through this hypothesis is that the aggressive tumors in glioma are likely associated with this particular aggregator of mitochondrial proteins. And ChatGPT is doing a really decent job with explaining that hypothesis. So this is the paragraph. I'm not going to read to you, but it really finds the connection between the two papers. So now I'm going to switch gears to the second project that I want to mention. It's called D2H2, Diabetes Data and Hypothesis Hub. And in this website, we aggregated tools that give you the ability to query single genes, gene sets, and we also did curation of diabetes-related publications in the GEO from bulk and single-cell RNA-seq as well as microarrays. So on top of all of those tools, we built this chatbot that gets input from the user. It can be a free sentence that the user composed, and then it finds the tools to best match the query and direct the user to the results. So why do we need this D2H2 website in the first place? So there are a lot of type 2 diabetes studies that are not being used. They're also buried in GEO and the literature, and diabetes researchers rarely go to them. And there are a few diabetes-related portals, but they are all aggregating data without an easy way to form hypotheses. And most of those portals are focused on genetics and genomics. And here is a graphical abstract of this project. We have tools that either query a single gene or gene set, and then this is combined with data sets, curated data sets. And all of those are fed into this chatbot that helps with hypothesis generation. So the user submits a query, and then the query is converted into a JSON format together with a list of tools that via their APIs, the OpenAI API need to make a decision how to combine the user query with the available APIs that can process the data. So for example, if the user enter a gene, it parses that parameter and it uses that as a part of the query into the right tool. And then you get rendered results, including figures, interactive figures, as well as links to the original analysis database. So this is an example. For example, you can ask the bot in which tissues STAT3 is expressed. It knows that there are several databases that have that answer. So it prompts the user for selecting one of the databases that have that information. And then if you click on the hyperlink of the database of your choice, here I clicked on ARCHES-4, you get a diagram of the expression of STAT3 across the tissues that are provided from ARCHES-4, as well as buttons to go directly to the sites that you can get more information and similar diagrams from GTEx and this new tool called Gene Ranger, as well as ARCHES-4. So the last part is explaining this idea of converting L1000 data to RNA-seq-like. And I think Swetab already discussed the LINCS L1000 data. If you don't know much about it, it's an assay that only measures 978 genes, but it can be done in high throughput. So there are about 4 million samples that were profiled with this assay for profiling the activity of 30,000 drugs on about 80 human cell lines where the drugs are applied in different concentration and time points. And then gene expression is measured before and after the drug treatment. And this helps you to have a database for drug discovery. You can put in your transcriptomics data. And then through the search of the up and down genes, you can find mimickers and reversers in this LINCS L1000 database. And the issue here is that most transcriptomics are done now with RNA-seq. So the L1000 and the RNA-seq are not really aligned. So there's two different assays and they don't really match. We also don't have all of the measured genes. So there's different ways to improve the imputation of the rest of the genes. We do, however, have 4 million L1000 samples and about at least 1 million RNA-seq, bulk RNA-seq samples in GEO. So we have a lot of unmatched paired data. So what we've done is that we use CycleGAN, which is a supervised learning method for generative AI for unpaired data. So when you have a lot of, let's say, images that are unpaired, you can learn how to convert the style of one image to another, or convert zebras to horses, or summer to winter, based on the training of many example. And this was done for images, but the idea is that we can apply this same approach to biological omics data. So the model is a two-step model. The first part converts L1000, the 978 L1000 genes to their RNA-seq-like 978 genes by taking random L1000 samples and random RNA-seq samples and trying to make them look alike using a CycleGAN. And the second part of the model, take the converted RNA-seq like L1000 and also the known RNA-seq data and try to develop a model that imputes the rest of the genes. And we have a better coverage than what is currently available for the L1000. And this work was done by Minji Jeon, who now has her own lab at the University of Korea. This is a visualization of some of the L1000 data that was profiled both with the L1000 and the RNA-seq. So the original data, you can see that the points are very separated, but after doing this transformation, we can bring the data to be more aligned and have the same structure, at least at the global level. The data is served for search through this aperture that allow you to put in a gene, and then you find drugs that may upregulate or downregulate the expression of the gene based on the data from the L1000, those 30,000 compounds, and the full set of the L1000 data. And also you can download the data in different formats. There are also CRISPR knockout data, overexpression perturbations, ligand perturbations, antibody perturbations that are also converted into this format from the L1000. And you can also get the model itself. So we give the model for free. You can download it, and then you can transform your own data into the RNA-seq-like format. So just to summarize, I showed you 3 new applications of generative AI for ongoing project in the lab. Ramagene is this mining of gene sets from supporting materials. Type D2H2 is this database for diabetes hypothesis generation. It has this chat interface on top of many of the tools that we have developed, as well as published data sets that are curated, and also this transforming of L1000 profile to RNA-seq-like. I would like to make just some general comments. Generative AI needs to be used responsibly. So in all the cases that I showed, we really limit the capability of the AI to do very certain things that we predefined, and basically making choices between various discrete answers. So it's sort of like giving a calculator to a kid. So the calculator cannot make any errors, and the kid may not understand the question, but it cannot make mistakes. In general, I think biologists are very skeptical of using any of those tools, so it will take some time to get it adopted, even though those new resources are very powerful. And this is only the first wave of generative AI. The capabilities of those apps right now, they're a little clunky, but they are improving rapidly. So finally, I would like to thank the members of the lab that did all the work. Daniel Clark was really leading the Ramagene mining and analysis together with Giacomo Marino. The D2H2 project is led by Giacomo Marino and assisted by Nashiat Ahmed. And then the L1000 to RNA-seq-like was led by Min Jijian, but Sherry Xie is also involved in the project, and she did a lot of the work in that for that project as well. So thank you so much for the opportunity to present to you today, and I'm happy to take any questions.
Thank you, Dr. Mayan. It was an excellent presentation. You got some applause going on in our reaction section here. We did have one question come in the chat, and it's about that D2H2 example. So it's from Ayush here. It says, extending on the D2H2 example as a concept, is there a possibility of building disease or context agnostic chatbot engines in the future, or will we have individual context or disease specific chatbots only?
Yeah, I think it's definitely, that's actually something that we are working on, is making it more general purpose. And it's really about the APIs that are sitting on top or below the chat bot. Mm-hmm. So absolutely, I think it's already somewhat disease agnostic, because many of those, and the example that I showed is also disease agnostic, but definitely that's sort of like the next step. The reason it's just diabetes is because this is affiliated with a grant that required us to make it diabetes focused.
Okay, awesome. If you have any more context that you want to add in or any comments, feel free to type them out or join the chat. Thank you again. It was a really awesome presentation. We appreciate you taking the time to speak with us.
Yeah, thank you so much. I'll be in the chat and I can answer some questions in the next few minutes.
Awesome. Thanks.